Good afternoon, madam. I think he has joined. Dr. Anand has just
Hi.
joined. Yeah, now you take up the matter now. Madam, you can start the program. Okay.
Shall I start?
Yeah, yes, please.
Yeah. I request our technical team please stop so much or comment. A very good evening to one and all. I have great pleasure to introduce myself. I am Dr. A. Sunita, Vice Principal, Head Department of Pharmaceutical Analysis, working since 2001 at Hindu College of Pharmacy, Guntur. As convener of today's program, I am happy to greet all of you here. On behalf of Hindu College of Pharmacy Management, staff and students, a genial welcome to one and all for a motivational talk and presentation on latest advances in novel drug delivery systems. Now, I request Mrs. G. Indira Priyadarshini to welcoming the delegates.
Thank you, ma'am. A very good evening to the galaxy of intellectuals present here. My name is Indira Priyadarshini. I am assistant professor in Hindu College of Pharmacy. And I got the privilege to welcome you all to this fabulous session of exchanging knowledge on latest advances in novel drug delivery systems organized by Hindu College of Pharmacy. Today is a special and memorable occasion, one that to be remembered to the years ahead. The reason behind is none other than our speaker, Dr. Ananth S.V. Pannalagaru. He is the live example for the saying, the quality of man's life is in direct proportional to his commitment to success. No synonym of excellence can completely explain him. We are honored to have such a great person here with us. I wholeheartedly welcome you, sir.
Thank you. Thank you.
A dream doesn't become reality through magic. It takes sweat, determination, and hard work. It is a belief endorsed by our management for the success of Hindu College of Pharmacy, which has become synonymous for academic excellence, a benchmark to measure the co-curricular excellence, and also a center which creates responsible citizens. They are always been a source of inspiration and motivation. On this context, I would like to introduce our beloved chairman, a man with gracious heart, professionally is a renowned advocate, Sri Jupudi Rangaraju Garu. I also want to introduce our admirable secretary and correspondent, Dr. S. Matsuran Rao Garu, who is a doctor by profession. I extend warm welcome to our principal, Dr. R. Govindarajan Garu, vice principal and program convener, Dr. A. Sunita Garu, PharmD director, Sri P. Sitaramai Garu, NAC coordinator, Dr. M. V. Nagbhushan Garu, co-convener of this program, Dr. K. Rajkumar Garu, all the faculty members of Hindu College of Pharmacy, and dearest students, welcome you all. I'm glad and thankful for giving me this great duty of welcoming. Thank you, one and all. I request Dr. A. Sunita Garu, program convener, to preside over. Thanks once again.
Thank you.
Thank you, Mrs. Indira. Now the program starts with lighting of lamp. I request our technical team.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा
Hindu College of Pharmacy is a temple of learning, and we are making our best effort to give quality education to our students. Hindu College of Pharmacy is a dream envisioned and realized by able administrators Sri Jupudi Rangaraj Garu and Dr. S. Madhusudan Rao Garu, dedicated to the mission of spreading education. Through their diligence and inspiring leadership, they have helped create a learning environment that represents a unique blend of distinguished faculty, brilliant students, and proactive collaborations with industry. We, the members of Hindu College of Pharmacy, bow our heads in respect to greet today's speaker, Dr. Anant Pannala Garu, a man with an outstanding intellect, eminent scientist, masters in NDDS, recipient of confidence, moreover, senior principal lecturer, School of Pharmacy, Biomaterials and Drug Delivery Research and Enterprise Group. We delighted to offer a rapturous welcome to Dr. Ananta Pannala Garu in this prestigious moment. It is the most memorable day with full of joy. I extend a warm welcome to Dr. Shankar Prasad Garu, the man who is continuously supporting to the Hindu College of Pharmacy. It is a very good opportunity for formulation scientists to overcome the many challenges associated with current drug therapy, thereby improving the management of patients in future. The advent of novel drug delivery systems and their application to almost all diseases require pharmacokineticists to rethink what data analysis and modeling approaches will be most useful. Before introducing today's speaker, now I request our Honorable Chairman, Sri Jupudi Rangaraj Garu, a renowned advocate in Guntur city, incredible human being, to address the gathering. Over to Chairman, sir.
నమస్తే
నమస్కారం.
నమస్తే
నమస్కారం.
నమస్కారం సార్, చాలా సంతోషం. మీరు తెలుగువారు అయి ఉండి యూకేలో చాలా అత్యున్నతమైనటువంటి పొజిషన్లో ఉండి మిమ్మల్ని ప్రస్తుతానికి కోవిడ్లో భారతదేశానికి వెల్కమ్ చేస్తున్నాను దీని ద్వారా. అందులో మరి గుంటూరు ప్రజలకు చాలా సంతోషం. I'm really very happy, sir. Our Hindu College of Pharmacy is established about more than 20 years back, and we are doing the best what that is possible. We have got good reputation as far as the academics are also concerned, and we have got good faculty. About a couple of years back, the then chairman, Dr. Manoj Krishnamurthy Garu, and myself, being secretary correspondent, we intended to start a research center in our institute. We made a little of effort, but we could not fructify in that aspect. But now we have got a good a guiding factor from you, who would try to put forth your views so that we could develop our Hindu College of Pharmacy also as a research center with your kind help and your benevolent view, so that in Guntur district, our Hindu College of Pharmacy would have a good center for this. I have got the good support of our secretary correspondent, Dr. S. Madhusudan Rao Garu. In fact, our board of directors have got eminent doctors, sir. Our honorary chairman is Dr. Manoj Krishnamurthy Garu, who is PCI member, and Dr. Vijay Bhaskar, an orthopedician, our vice chairman, and our Madhusudan Rao Garu is one of a good, wonderful doctor with good hand. And the other one is Dr. R. Ramakrishna Garu, who is also a chest physician. So Hindu College of Pharmacy is embroidered with the knowledge that is required as far as the pharmacy is concerned. When I have gone through your biodata, sir, I felt that I am talking to a wonderful personality who is doing his human service to the human mankind and especially to the students also. I felt that whenever you come to India, it is my bounden duty to ask you to come to our institute, put your good wishes, and also help our research center in this respect, sir.
It will be my
Apart
pleasure.
from this, Shankar Prasad Garu, he is also our well-wisher. I was told that he is none other than your brother-in-law.
Yes.
So, when Shankar Prasad Garu is very close to us, that means you are also bound to be close to us. That is I am putting it in a legal way, not in the other way at all. And పన్నాల అనగానే నాకు అనిపించింది అసలు మీరు మీ యొక్క చదువే పన్నాల, మీ యొక్క ఆలోచనే పన్నాల, మీ యొక్క క్యారెక్టరే పన్నాల. మీరు ఎంత పన్నాల వారు మాకు కూడా ఈ కాలేజ్ ఇన్స్టిట్యూట్ సహాయం చేయమని చెప్పి మిమ్మల్ని అభ్యర్థిస్తూ ఐ విష్ all because I must be under to you. So I feel that God should give you all grace and see that with all good health, with your family, especially in this COVID, which is yorking us, this yorkingness of human life. We do not know where it goes. The pandemic is creating a lot of havoc in the entire human hemisphere. And I pray that Lord Dhanvantri should give his all blessings to the human mankind and see that this human mankind is survivable. Thank you, sir.
Thank you.
And I hope our students and all of us will get educated by your good lecture, sir. Thank you, sir.
Thank
Namaskaram.
you very much. Thank you. Namaskaram. Thank you.
Thank you, sir, for your blessings and crisp message. It is indeed a great pleasure to introduce today's speaker, Dr. Anant S.V. Pannala Garu. I request our technical team, please share the screen. Yeah. Sir, it gives me immense pleasure to introduce today's speaker, Dr. Ananta S.V. Pannala Garu, Principal Lecturer, School of Pharmacy and Biomolecular Sciences, University of Brighton. Coming to his educational qualifications, Sir has completed Bachelor of Pharmacy from College of Pharmaceutical Sciences, Manipal in 1991. He did Master of Pharmacy in Pharmaceutical Chemistry from College of Pharmaceutical Sciences, Manipal in 1993. He was awarded with a Doctor of Philosophy in Chemistry from Department of Pharmacy, University of London in 1997. Never contented in his research for knowledge, he also conquered postdoctoral fellow from King's College London, UK during 1998 to 2003. Apart from his studies, he is a member in various professional affiliations and trainings like Senior Fellow Higher Education Academy in June 2019, Research Fellow of Royal Society of Chemistry in 2017, Member of the Royal Society of Chemistry in 2008, Prince II Project Management Training Foundation and Practitioner in 2008. Served as Study Director in GLP in 2004. Served as Member of Controlled Release Society, Member of Indian Pharmaceutical Association, member of Academy of Pharmaceutical Sciences, member of American Association of Pharmaceutical Scientists. Coming to his work experience, Sir has vast field of experience as a trainee industrial pharmacist in the year 1990. Part-time teaching in King's College London during 1994 to 1997. Sir served as a visiting scientist at Tech Hochschule Zurich, Switzerland in 1998. Sir did postdoctoral research fellow during 1998 to 2003, worked as a principal scientist from 2003 to 2005, and served as head of formulation and pharmaceutical analysis from 2005 to 2008. He worked as senior lecturer from 2008 to 2013. Presently, Sir is working as adjunct professor of pharmaceutical sciences from 2007 at School of Pharmacy and Biomolecular Sciences, University of Brighton, United Kingdom. Coming to his achievements, his scholastic achievements engendered very active involvements and leaderships in countless colleges, professional organizations, and universities across the world. His enormous experience in research helped him to guide various research projects. Sir has guided 83 research projects at PG level, out of that 35 MSc students, 48 MPharm students, guided 5 PhD scholars as a main supervisor, 1 international MPharm project in collaboration with Manipal University. Sir also guided 6 research projects at UC level as a main supervisor at University of Brighton. In addition to that, Sir had accomplished various achievements like organized 2 Royal Society UK funded international conferences in Manipal University, India, established a new master's course on industrial pharmacy in University of Brighton in 2010. Co-developed framework for new postgraduate courses in the University of Brighton. Implementation of new analytical process for better efficiency in the analytical laboratory, Medpharm Limited. Cost saving in analytical equipment, procurement for Medpharm Limited through negotiations with vendors. Not only that, Sir lead to a grant from Ministry of Agriculture for PhD work, Fisheries and Food Agency for his first postdoctoral position. Invited as a speaker at Second International Conference on Food Factors, Chemistry and Health Promotions, Japan in 1999. Overseas research scholarship during 1994 to 1997 funded for PhD program. Presented his MPharm thesis at 5th INVIN WHO conference in Geneva, Switzerland in 1993. Coming to his credentials, distinction was inseparable from his studies as he constantly received numerous citations, scholarships, and various prestigious awards. Sir has published 33 papers in various international journals with 22,481 citations. Sir, it's really amazing and amazing. Over the years, Sir has attended across 32 conferences around the globe. Sir has reviewed many journals and books. We are very fortunate enough to have such eminent speaker to enlighten us. With all my affable greetings, I cordially invite Dr. Anant S.V. Panalagaru to deliver the lecture. We are so thrilled to hear you, sir. Over
Thank you.
to speaker.
Thank you very much for your kind introduction, madam. I'm really humbled with your introduction. Um, before I start, I would like to say thank you to the chairman, sir, Sri Rangaraj Garu, also Shankar Prasad, as well as everyone else in the team for inviting me. It is really a great honor for me to be giving my talk. Um, I do not know much about being a motivational speaker, but what I will try to do is what motivates me. So I'm going to talk more in that and what motivated me to be here today this far, how I have come this far to talk, to be able to talk about my research. So that's what I'm going to do, if that is fine. What I have done is I planned for about 20 minutes to about half an hour roughly about my talk, and then I will take any questions. So should I just share the screen, or I've never used Zoom to do any presentation, so this is my first time, so please bear with me. My camera also just tries to switch off by itself. So if it does happen, then
Okay.
I'll try to connect again. Um, how do I share screen again? Oh, okay.
Yes, sir, it's connecting.
Which one is connected? Can you see my presentation
Drug
or?
delivery. Yes, sir. Drug delivery systems.
Okay. I'm sharing this particular screen. Okay. So I need to move it across. Hopefully started.
Yes, sir.
Yeah.
Yes, sir. Sir, please disable participants annotation tool. It is in more option in the Zoom.
Where is that?
In more option, disable the participants annotation tool.
Disable participants annotation. Okay.
Yes, yes, sir.
So,
You please
so
proceed.
that's fine. Okay.
Yes, sir.
Okay. What I have changed the title slightly, and I'm going to talk about drug delivery systems, the trials and tribulations that I have faced in my research career. But before I do that, what I wanted to say was what made me go into research. So this was a quotation that I came across in 1991. This was in my professor, Dr. Amenera in Manipal. And he had this particular quotation as a small poster. And that kind of made me think a lot more about research. There is one thing even more vital to science than intelligent methods, and that is sincere desire to find out the truth, whatever it may be, and then advance. So this is a kind of thing like it doesn't matter whether you get a negative result or not, whether you get a positive result or whether you're using a state-of-the-art technology. It is basically what is it that we are trying to do and understand more and then move on from there. So that was my first inspiration to start my research. You've already given everybody my detailed background, but I thought I'll add a little bit more to it. During my MPharm, what I did for my project was I synthesized this particular molecule. It is a curcumin analog. It was a new chemical. It was a new molecule, and we managed to get some really incredible results. And that I managed to present in as an MPharm student from India, I managed to present this work in a WHO conference in Switzerland. And that was the next thing which motivated me to apply for a PhD and join King's College London for my PhD career. And that's where I took these natural products, because curcumin being a natural product, I'm going to talk more about curcumin a little later on, and also tell you about why I'm talking about curcumin. So that kind of developed me to, motivated me to increase my interest in natural products and how it is actually beneficial in human health. One of the main things was, we always say, eat green fruits and vegetables. Being a vegetarian has got a lot of beneficial properties, etc., etc. And all our Ayurveda, it says that also. So why does it say that? How does it actually interfere? So that was my PhD project. So basically what I was looking at was the oxidative stress in a disease condition, such as a cardiovascular disease. And then looking at these natural products, that is the flavonoids, and how they help in reducing these situations, such as how it actually reduces the atherosclerosis, or the plaque which we find in the arteries that leads to heart attack, stroke, etc. Fine, it is easy to say that eat more fruits and vegetables, but as a pharmaceutical scientist and as a pharmacist, one would think that yes, it will be nice if you are actually able to get these chemicals out and give it as a medication. So how do you do the delivery? But before that, what I did was a little bit of clinical research experience, where I looked at, do they actually have any beneficial properties at all or not? So that is where I looked at as a postdoctoral research fellow in King's College London. And I was working at these two hospitals, Guy's and St. Thomas's and Royal Free. Guy's and St. Thomas's, by the way, was established in 1700. So it is one of the oldest hospitals in the world. And also the two main discoveries. One was the DNA. The second was how the cancer happens, the Hodgkin's disease, how this happened. They were both discovered in the same lab as where I was working at Guy's Hospital. So that kind of being in a place that where such excellent science had happened motivated me even more. So I've looked at further in these natural products and looked at how beneficial properties, etc., could be done. After that, I had a brief stint in the industrial setting where I was there for about 5 years. And I was looking at development of new formulations for different drugs. So they were all brand new drugs, and they were all established or developed by other companies. And we wanted to see whether we are able to develop the formulations for giving it by transdermal route, that is application to the skin, or pulmonary route, that is by direct inhalation. Drug delivery through the nail, apply it onto the nail and then see whether it actually goes through by injection, that is parenteral drug delivery, or also by vaginal and rectal drug delivery route as well. So we're looking at different ways of administering these drugs. So since 2008, I have been in academic. I've been in academic and I've been in research. That's where I've had 6 PhDs so far, and quite a number of MPharm and MSc research projects. Majority of my PhDs and MPharm projects were all based on drug delivery, on pharmaceutical technology, and how we can actually look at developing this new technology for a particular condition. So what exactly are we trying to do in terms of a research area, especially in drug delivery and research? Possibly in the olden days, where be it Ayurveda, be it Unani, be it the old Western medicine, doesn't matter. Whoever it was, they used to go to the doctor, the compounder, the apothecary, whoever it was, and say, this is the condition, and they'll look at it. తెలుగులో చెప్పాలంటే నాడి ముట్టుకొని ఇది ప్రాబ్లం అని take a little bit of whatever extract and then shove it down the person's throat. Problems. First problem, you do not know how much dosage you gave. Second problem is you do not know whether the drug has actually reached the site of action. Third problem is the patient is very much likely to spit it back onto your face. ఎందుకంటే మొత్తం అన్ని మెడికేషన్స్ చాలా చేదుగా ఉండేవి, అదొక పెద్ద ప్రాబ్లం అయ్యి. So when you're developing a formulation, when you're developing a drug delivery system, what you got to look at are these 4 basic points. First is ease of administration. How easy is the drug to be administered to the patient? Orally is the easiest way. Alternatively, we'll look at other ways if it is possible. Why oral is the easiest? Because it's just like eating anything. Ask the patient to take it whenever they want to, whenever they have to, and they'll be happy to do that. They'll be happy to go about their own way. If you say you have to go and see the nurse or the doctor and take an injection 3 times a day, no chance. So that's a problem. Controlled dosage. So if you say it is 500 milligrams of paracetamol, it is exactly that much. You're not going to change anything. Increased efficacy. So you can make sure that the exact amount of drug reaches the site of action, and reduced toxicity as well, because you'll be able to control the exact amount that needs to be given, and the doctors can change the dosage, the amount that needs to be given. Maybe it's not 3 times a day, maybe 2 times a day or whatever. So the main concepts as a formulation scientist that one needs to remember are these 3 concepts. First is patient compliance. Second is design the drug delivery system for the drug, not the other way around. I'll explain this a little bit more in a few minutes. And then go through the actual dosage form development. When I say patient compliance, what exactly does it mean by patient compliance? ఇక్కడ నేను కొంచెం తెలుగులోకి ట్రాన్స్ఫర్ అవుతాను, and I'll talk about this one. అందరు చాలా మంది చూసే ఉండొచ్చు ఈ సినిమా, మిస్ అమ్మ 1955లో వచ్చింది. అందులో అల్లు రామలింగ గారు పంతులు దాంట్లో పనిచేసేవారు. ఆల్సో హీ వాజ్ ఆయుర్వేదిక్ డాక్టర్. అందులో ఒక పర్టికులర్ సీన్ ఉంది. ఆ సీన్లో ఏంటంటే సావిత్రికి ఒక దయ్యం పట్టినట్టుగా యాక్ట్ చేస్తుంది. సో ఇప్పుడు దాన్ని ట్రీట్ చేయడం కోసం అని చెప్పి అల్లు రామలింగ గారిని పిలిస్తే, పంతుల్ని పిలిస్తే, ఆయన వెళ్ళి నాడి పట్టుకొని చూసి, ఇందులో వాతాహరిణి మాత్రం ఒక 2 పాళ్ళు, పైత్యం మాత్రం 3 పాళ్ళు తేనెలో రంగరించి కలిపితే తెల్లవారే కల్లా తగ్గిపోతుంది అంటారు ఆయన. ఆ వాతాహరిణి మాత్ర, పైత్యం మాత్ర అంటే ఏమిటో నాకు తెలియదు, కానీ ఒక పర్టికులర్ పాయింట్ మాత్రం ఉంది, తేనెలో మిక్స్ చేసి. వై ఇస్ దాట్? That's because to make these 2 tablets palatable. ఎందుకంటే తేనెలో తీయగా ఉంటుంది కాబట్టి ఇచ్చేసారు. ఇవి చేదుగా ఉంటే ఎవడో తినడు. అలాగే ఈ టేస్ట్ గోంగూర పచ్చడి టేస్ట్లో లేకపోతే గుత్తొంకాయ టేస్ట్లో ఉంది అని అంటే కనుక లొట్టలేసుకుంటూ వేసుకుంటాడు, కానీ పేషెంట్ చేదుగా ఉంది నాయన కాకరకాయ ఎలాగా అంటే తీసి పక్కన పడేస్తాడు. సో that is the main thing which we got to look out for, that the patient is happy to take the medication. So as a formulation scientist, that is the first thing that we need to look out for. What is patient compliance? Doctors దగ్గరికి వచ్చేసరికి పేషెంట్ కంప్లయన్స్ అనేది కంప్లీట్లీ డిఫరెంట్ ఆస్పెక్ట్. వాళ్ళు వేసుకుంటున్నారా లేదా, సరైన అమౌంట్ వేసుకుంటున్నారా లేదా అన్నది వేరే చూసుకుంటారు. దాని గురించి కాదు మనం యాజ్ ఫార్ములేషన్ సైంటిస్ట్ వీఆర్ నాట్ వరీడ్ అబౌట్ దాట్. వాట్ వీఆర్ వరీడ్ అబౌట్ is the patient going to take it or not? That is the first step. The second one is understanding the drug. ఇందాక ఒక చిన్న ముక్క అన్నాను, గుత్తి వంకాయ కూర అని. సో సపోజింగ్ ఆ గుత్తి వంకాయ కూర అనే ఒక టాబ్లెట్ అనుకుందాం, అది ప్రిపేర్ చేయడానికి ఫస్ట్ అర్థం చేసుకోవాల్సినది ఆ వంకాయ ఎట్లా ఉంది అని. అది గుండ్రం వంకాయ, పొడుగు వంకాయ, గట్టిగా ఉందా, సాఫ్ట్గా ఉందా, ఏం చేస్తుంది, అందులో ఎటువంటి మసాలాలు వేయాలి అన్న సంగతి తర్వాత పట్టించుకుందాం. ముందు దాని గురించి అర్థం చేసుకోవాలి. అలాగే ఏ కూర తీసుకున్నా కూడా మీరు ముందు ఆ కూరను అర్థం చేసుకున్న తర్వాతే మిగతా మసాలాలు, ఈ పొళ్ళు అవన్నీ వేయచ్చు. Exactly the same principle you apply for a formulation development, where you look out for understanding the drug. That is, we call it as pre-formulation. So for example, if we take curcumin, which is the active ingredient in turmeric, so understand its particle size, crystal properties, solubility. Is it water soluble? Is it non-water soluble? And oil soluble? Does it have any chargeable groups? What is it log partition coefficient? That is, how much of it is oil soluble? Once you administer it, how much of it is going to be bioavailable? That is going to be a direct indication of log P. And of course, you have the organoleptic properties such as the sight, smell, taste, etc. So all these things are going to play a predominant role when you are trying to understand the drug and understand it's what the actual formulation that we are going to develop is. So this is the first stage, and doesn't matter what type of formulation we are preparing, we have to do this test for the drug, for all types of dosage forms and for all drugs. So as I said, I'm going to talk a little bit about about curcumin itself. Curcumin is the active ingredient in turmeric, pasupu. And if we look at our Ayurvedic system, it's thousands of years old. It has been used for a variety of conditions. And if you look at the number of publications that are there today, per year, there are at least 10,000 new publications that come out on curcumin alone. So be it in their activity, be it in its activity, be it in its formulations. So if you look at these curcumin formulations, you have different types of tablets that are available, you have capsules, and you have a powder that I'll come to a little bit later on. And these are also capsules. So this is actually a syrup on the bottom right. And apart from that, these are all oral dosage forms. Apart from that, we have these advanced dosage forms, such as these polymeric conjugates, solid lipid nanoparticles, micelles, liposomes, nanogels, etc. So there are a number of advances which have actually been tried out for delivering this particular drug. If you look at the chemical properties of curcumin, it exists as two separate forms. First is the keto form, and that's the enol form. And depending on the pH of the solution, a particular form predominates. But the main thing which we got to remember is you only have about 11 nanograms per milliliter in pH 5 buffer. And apart from that, even if you administer 12 grams, so if I were to put that into context, if you were to take 24 crocin tablet size curcumin tablets, you only have about 10 nanograms that will be available in the blood. Majority of it goes through extensive first pass metabolism and is excreted. In spite of that, it has very good properties, very good pharmacological properties. It is useful for various conditions. It is a good antioxidant, known to be a good anti-inflammatory. It is also shown to be an anti-cancer drug, gastroenteric systems, different gastric problems. So it's quite a good number, but nobody has been able to solve this particular problem of poor solubility, and how is it going to be, how we can actually increase that. So how do you overcome that, the poor bioavailability? There are three simple ways which you can do. First is Easy to say, increase the rate of dissolution, increase solubility, and increase stability. If you're able to do any one of those, you will be able to achieve that. So if we look at the administration, oral administration of curcumin, so the patient just swallows a tablet or a capsule. The curcumin particle goes straight into the stomach, and that's where it dissolves. And then slowly the gastrointestinal fluid, a maximum amount is there, and it is absorbed and it goes into the bloodstream. But majority of it is going to be metabolized by the liver. If you were to look at alternative ways, say for example, if you are able to say give a maximum concentration that is available, that is a concentration that is greater than the maximum curcumin solubility, then in one way you will be able to increase the absorption. So you're actually giving a very high concentrated dose, but in a very small amount. Okay, very small amount administered, but maximum solubility. And that's where you might be able to increase that. And that's what we try to achieve by using this particular technology. It's a very, very old technology, possibly around 35, even more than that, about 45 years old. It's called as ordered mixing. One is where you have something which is water soluble, such as lactose, and you put curcumin on top of these lactose particles. So when you mix it continuously, the curcumin particles adhere onto the lactose. And once you administer it, once the patient swallows that, the lactose, because it is water soluble, just dissolves, and curcumin is absorbed from the gastrointestinal tract. Okay, so this is the way that we have administered this particular drug. Something else has come up on my screen about someone raising a hand, and I'm not able to control my screen anymore. Oops. There it is. Okay, fine. I got it back. So there are, as far as the hand raising, if there are any questions, I'm sure that we'll be able to take it at the end of the presentation.
Yes, sir. Yes, sir.
Okay. Yeah, so that should be fine, right?
Yeah.
So the latest advances in the drug delivery systems include where you have biopolymers which are used, and where you incorporate the drug within the biopolymer, or you have liposomes which carry the drug. Again, I'll go through it a bit more in detail in subsequent slides. And you have cyclodextrins, which are cage-like molecules. So you trap the drug. So the cyclodextrins are the ones which are water soluble, but inside is actually very hydrophobic. So it absorbs the drug and it carries it through. So it increases the bioavailability. And then you have dendrimers, which are simple repeating chain molecules that are actually quite useful, especially when you're delivering the drug. Gold nanoparticles, again, these are one of the hot topics now. We have developed some liposomes where we've combined gold nanoparticles with liposomes and then used the drug for administration. That actually is very, very useful. And micelles, which are simple surfactants that can be used. Carbon nanotubes and quantum dots are, again, which are mostly used for delivering non-organic molecules for drug delivery. So the basic thing which we got to understand, especially when it comes to nano size or nanotechnology or microtechnology, is first we need to know what is particle size. So if you were to take large particles, for example, even whether it is grain of rice or wheat, they're actually quite large particles. You break it down, then you have coarse particles, such as say coarse semolina, like Bombay rava, which is quite coarse. and then you break it down even further, like maida, that would be around 1 to 5 microns. So that's a very, very small particle. You break it down even further, then you have submicron particles and then nanoparticles. And nanoparticles, by definition, is you can have it as any length, but one dimension of it should be less than 100 nanometers. So it can be a micrometer long, but if its width is less than 100 nanometers, then it is defined as a nanoparticle. Again, I have a few examples here. That is just the coarse lactose that is used for, I don't know whether I'm still there. Am I there, or are my screen gone, my camera gone?
Yes, sir.
Can anyone tell me whether it is
We
it
are
working?
able, it's not visible, sir, but we are.
You're able to hear me, right?
Yeah, yeah.
Okay, I'll fix my camera in a few minutes. But there
Yes,
it is.
sir.
Yeah, that's fine. Sorry, I'm having a bit of a problem here. So what we have here on the left is the lactose particles that are commonly used for preparing tablets or capsules. The one on the right is actually a cell model where we've looked at the incorporation or uptake of these particles in a cells. These are scanning electron microscope images, these three. And these are microparticles and these are nanoparticles. Again, I'll go through a little bit more in detail in a few minutes. So if you were to look at where are we going to use the nano or micro technology? They can be used as a lab on a chip, for example, say on the diabetes strips. That's based on a nanotechnology. But what we are interested in is how can we actually incorporate the drug inside these nanoparticles, and how can we actually administer them. So what type of drugs can be used? You can have a various range. Corticosteroids such as budesonide and fluticasone, both are used for asthma and COPD treatment. Antibiotics such as levofloxacin, nucleotides such as siRNA, antifungal agents such as voriconazole, and of course, even the latest ones are also being tried, especially in anti-cancer and antiviral drugs. That is the one which they are trying to test out on for various viral infections. Not sure about COVID yet, but I'm sure that somebody will come out with something using these particles with one of these antiviral agents. And of course, you also have the diabetes insulin also, which has been administered. So what happens when you incorporate a drug into a nanoparticle then? First is if you take a dendrimer, you have the core unit. So this is where these are the ones in the pink are the repeating units, and the drug can either be attached to the on top of the surface ligand, or incorporated within the dendrimer unit itself, or within a micelle where you have the hydrophobic core and then the hydrophilic heads here. So you have the drug, so which is oil soluble. So if you have something which is oil soluble, that can go inside this particular micelle. And then you have liposomes, which have a hydrophobic core here, and then there's a hydrophilic center, and of course the outside is also hydrophilic. So you can, depending on whether your drug is oil soluble or water soluble, the drug can be either in the middle or within this core, or within a shell nanoparticle where you have a matrix nanoparticle where the drug is dispersed within the matrix, or you have the drug and then it is covered within a biopolymer on the outside. So there are different varieties that we can use. But why do we want to encapsulate the drug itself? First is protect the drug from degradation, especially if it is susceptible to extensive stress or extensive metabolism. You're able to protect the drug by encapsulating it, by putting it into a protective shell. And that, because you're also reducing the particle size, this can actually go through the membranes and into the systemic circulation quite easily, because of the cell width that we are between the cells that we are looking at is extremely small, and these nanoparticles can easily go through that. So you're also able to modify the pharmacokinetic profile. For example, say you can target straight to the area of action. For example, you have a tumor. You can put something which is on top, some like an antibody, and that can take the drug all the way to the tumor, and that's where it's going to break and release the drug. So you do not have the drug which is going all over the body, increasing the toxicity. It can go straight to the point of action. Okay. And I'm briefly going to talk about my research study that we have recently done. I've looked at co-encapsulation of tamoxifen, which is an anti-cancer drug, along with curcumin. So I've taken two drugs and put them together inside a PLA nanoparticle, that is polylactic acid nanoparticle. And this is for parenteral drug delivery. And the second one that I've used is as a microparticle using gelatin and gum acacia as the external core. So the microparticle can be given as an oral suspension. Why these two? Tamoxifen is an anti-cancer drug. It is well known, but it is non-specific. When you give it orally, it goes through all over the body, leading to extensive toxicity. Majority of the anti-cancer drugs have got the same problem. So it is not just the cancer cells, you're also affecting other cells to this toxicity. And it has been shown that curcumin, when administered with paclitaxel, has minimized the paclitaxel-induced toxicity. This was just basically simple mixing of both of them. It was not a nanotechnology-based formulation here. And they showed that, okay, this one, curcumin, reduces the paclitaxel toxicity. So we thought, if we were to combine these two and put it inside a nanoparticle or a microparticle, will we be able to see a similar effect? Can we actually reduce the toxicity of tamoxifen? And can we actually target the drug straight to the site of action? Where we have come so far is we have prepared these two products, we have characterized them, and we looked at the toxicity in a simple cell model. What I have on the left here are the PLA nanoparticles. The size of these particles, these are solid particles. So you have about 250 nanometer sized particles here with the drug in both the drugs incorporated inside. So this is where you have a matrix. So once it is administered, the PLA would slowly degrade and release the drugs and at the site of action. By this way, you're able to reduce the amount of dosage that you need to give it to the patient. Okay. The zeta potential is really important. That is the surface charge that is on these particles. That's about minus 22 millivolts. It means that if you were to prepare a suspension, because of the charge, these particles will repel each other and it will stay as a stable suspension. So that was quite useful. The second one is something which we have developed in my in my lab recently, is this solid microparticles. What you see in this eye shape is basically you have the solid gelatin gum acacia polymer. It is actually a conjugated polymer. And what you see as the eye inside is the liquid. As you know, curcumin and tamoxifen are both not water soluble. I've been saying that all through. So if they are not water soluble, by giving it in this way, we are again dependent on them being solubilized. What we have done here is we have solubilized it first in an oil, and we've incorporated that liquid, the oil, inside the solid capsule. So when this is given orally, it will go straight to the down the gut and immediately break down and release the oil. It's because they are already solubilized, they should be able to be absorbed much more easily. In fact, we have done this work with another corticosteroid as a proof of concept, and I have tested this in an animal model along with one of my students who was visiting from Manipal University, and she has shown that you can actually have a delayed release compared to a simple oral budesonide solution, as opposed to a microencapsulated one. Here, once again, the zeta potential being -18 shows that if you were to prepare it as a simple suspension, the suspension is going to be very stable and it is very easy to administer. And of course, you can make the suspension as palatable, etc., as required. So what we can conclude from the work so far is that we managed to incorporate both the drugs in these nanoparticles. We've seen some kind of a synergistic effect when we looked at it in a cell culture unit, especially the MCF7 cell line. So this is actual cell culture. These are cancer cells, breast cancer cells. And as future work, what we need to know is what is the rate of release of these drugs, and then formulate the drugs into an appropriate formulation, be it as an IV or as an oral drug. So that's how far I have come. So thank you very much for your attention. If there are any questions, I'll be happy to answer. Can I stop sharing?
Sir.
Should I stop sharing this? Yeah.
Yes, sir.
Okay, let me see.
Thank you very much, sir.
Thank you.
Sir, enlighten us the concept of drug delivery systems, factors to be considered, including patient compliance, pre-formulation studies, curcumin properties, bioavailability, enrichment of curcumin. And not only that, sir has covered advances in drug delivery, including liposomes, dendrimers, carbon nanoparticles, gold nanoparticles, etc. With this, and now I request Dr. A. Sita Devi, Professor and Head Department of Pharmaceutics, proceed questionnaire session.
Thank you, madam. Good evening, one and all. I am Dr. A. Sita Devi, Professor and Head Department of Pharmaceutics. Today, we are very much blessed to have Dr. Anantha Garu with us. The session was very impressive and highly informative, sir. We are very thankful to you and we expect a lot more guidance from you in the future. Yeah, now I'll move into question and answer session. Many of the people found the session is really interesting and had a few questions. Sir, shall I move to question and answer session?
Yeah, absolutely, sir? Absolutely.
Okay, sir.
Sure.
Yeah, first question is, which animal model best suits for evaluation of solid lipid nanoparticles?
It depends on the condition. Sorry, my camera's gone again. I don't know why. Oops. I'm sorry. Can you give me just 1 second?
Yeah, now it is coming, sir.
No, I think there is a loose connection. Give me 1 second. I'll just sort it out.
Okay, sir.
Uh-oh. Sorry, my camera fell off. There. Should be okay now, hopefully.
Yes, sir.
Yeah, sorry about that. Yeah, which animal model?
Yeah, best
Well,
suits
it
for
depends on what is the condition. So you don't choose the animal model based on the drug delivery system. You choose the animal model based on the pharmacological response that you are looking at. Because for any animal model, or rather, you can administer majority of the formulations to any animal model. So you look at what kind of response you are looking at, and depending on that, you choose the model. I would say that.
Okay, sir. Next question. How the bioavailability varies between normal drug and nanoparticle delivery, and its impact on PK/PD profile of drug.
Normal delivery and nanoparticle delivery. Let us say that you have the you're administering it by intravenous administration. We do know that if you were to give it as a simple IV infusion or IV injection, so you have immediately you see you have the clearance which is starting from the moment you have administered a normal formulation. If you have a nanoparticle, first you are hoping that the nanoparticle actually reaches the site of action first. And that's where you have the drug which is going to be released, and then the drug activity, and then the drug release. It depends on the clearance rate. One thing which we can know is by giving it when you're giving it as a nanoparticle, you're actually administering that as a suspension. So I would say that it depends on the clearance rate of these nanoparticles, how quickly they are cleared when they are administered. So that is a really important aspect that one needs to remember. Specifically, if you were to look at tamoxifen, for example, which tamoxifen is not a good example. It's given orally. I'm thinking of let's think of an antibiotic which is given by IV administration. That is given as a solution, and then it actually goes with the clearance quite quickly. But if it is given as a nanoparticle, you're possibly reducing the dosage that needs to be given, and also the clearance is going to be much slower compared to that. I hope that answers the question, but it's really
Yes.
difficult to say which way it would, because with nanoparticle, it's quite a difficult prediction that everybody is facing at the moment.
Okay, sir. Next question. How can we reduce first pass effect of oral route?
If the drug is susceptible for first pass metabolism, the only way that you can overcome that is by increasing it, or by first is prodrugs. Go for a different, change the molecule so that you can actually reduce the first pass metabolism. Alternatively, go for an alternative delivery system, such as a microcapsule, where you are actually protecting the drug, and the drug is only released from the formulation when the formulation reaches further down the gastrointestinal tract. One of the main things which we always say is for that to reach the ileum stage of the gastrointestinal tract intact, and only then, if it is absorbed, then you are likely to reduce the first pass metabolism.
Okay, sir. Next one, sir. What are some different methods used to deliver medicine into the body?
Oh, there are loads. How long is a piece of string? So right through from చెప్పాలంటే నశానం నుంచి పాదాల వరకు ఏదైనా తీసుకోండి, ఎటు పక్క ఎలా అన్నా ఇవ్వచ్చును మనం.
Yes, sir.
You can give it by oral route, you can give it by inhalation, nasal, through the eyes, skin, which is the biggest organ that is available for us to administer. But one thing which you must remember is what are the properties of the drug,
Yes, sir.
and depending on the properties of the drug, choose the dosage form. Don't pick a dosage form and put any drug into it. It will not work like that. Because if it is oral, we have to follow the Lipinski's rule of 5. So it has to have a certain number of hydroxyl groups. It has to have a log partition coefficient, molecular weight, etc. All these factors which are there. So you cannot give any drug orally. Similarly, you cannot give any drug which has got a very high molecular weight. For example, insulin, you cannot give it by topical because it's a very, very large protein. So you choose it based on that.
Yes, sir. Sir, next question. How to select a suitable medium for drug release studies? Whether it depends on solubility of the drug or GI environment into which it is going to release.
How to choose the medium?
Select, yeah, yes, sir.
It depends on where you want that to be. For example, are you looking at increased bioavailability? If you're looking at increased bioavailability, choose something which is much closer to the plasma. Even if you go for phosphate buffered saline, that would be a suitable medium. But you're again limited by how much of the drug is soluble in there. For example, if you go for an oral route, as the pharmacopoeial recommendation, majority of the dissolution studies, we always look at 0.1 molar hydrochloric acid for the dissolution. That's a bog standard accepted one. And then from there, we always alter, say, oh no, we want it to be in the ileum. Okay, change the pH. Go to pH 6.8 buffer and put it in the dissolution medium, and then you look at that. That is based on oral route.
Sir, next question, sir. What is the suggested dissolution fluid volume for colonic drug delivery? Fluid volume.
Fluid, it's difficult to say. I mean, because for colonic drug delivery, you're giving it orally.
Yes, sir.
And if you're giving it orally, the way that you're using is the standard pharmacopoeial method, which is if it is the BP, if it is British pharmacopoeial method, we use 0.1 molar hydrochloric acid, 900 milliliters for the tablet or the capsule, which is put in the basket or the paddle method. But you can actually look at a continuous flow dissolution method as well. And in that way, if you're going to use that particular method, you do not have a volume because it's a continuous flow. You have the drug which is held within a unit, and you're passing the fluid through the unit. So you're not. It is only this particular volume that is used, usually around a 50 milliliter chamber that is used for the continuous measurement.
Means
I
if
don't
you
know
want to
whether that
use
answered-
simulated colonic fluids, if we
Simulated
want to keep simulated colonic fluids, usually colonic fluid, the content is less than the gastric fluids.
Yes.
Yeah.
So what
Then
I wouldn't
usually
worry
we
about,
use 900 ml only.
Yeah.
Colonic contents are the fluid volume is less than the GI content, no, sir?
Yes, it is.
So yeah, then what is the suitable volume, appropriate volume?
I would look at rather than the normal dissolution method, which is where we use the paddle, the bucket, the mesh or the paddle method. I would choose the continuous flow through method, where you have a chamber. So you're not worried about the volume anymore, because it is going to be continuously changing, because it's excuse me, the volume is continuously being reabsorbed, and then it is excuse me, I'm just going to get a sip of water. So I would choose a I would choose one of the other models rather than a dissolution bath for that. And you preferably use but I know that colonic content doesn't have a buffering capacity, but I would still use around pH 8 or for the ascending colon, or pH 7 for that.
Okay, sir. Next question, sir. Which route has highest bioavailability?
Parenteral, IV.
Yeah. Yes.
Yeah.
And, another question, sir. Is topical gel helpful for migraine headache?
It depends on the drug rather than on the formulation. Migraine headache, you can sumatriptan was one of the drugs which was developed as a drug for the treatment of migraine. Imigran, oral spray, nasal spray, fast dissolving oral films. They were all used, but it was not successful giving it topically, because the drug would not go through the skin. So if your drug has got good log partition coefficient and it can go through the whole thickness skin, then yes, definitely that formulation should work. But then again, you have to optimize it in such a way that the formulation in such a way that the drug is happy to leave the formulation and go through the skin. That's the problem with every topical formulation.
Yes, sir. Another question, sir.
Yeah.
Can we use microneedle concept in transdermal drug delivery system?
Yes. Yeah. It's one of my colleagues works extensively in that. We're using drug which is impregnated on PLA microneedles, which are then put onto a patch, and that is administered straight onto the skin, and then the drug is slowly released across the. One thing which you're overcoming is the stratum corneum barrier, which is the main problem for drug absorption.
Okay, sir. Next, another question, sir. How can we avoid the dose dumping in case of transdermal delivery of insulin?
Transdermal delivery of insulin? I've not come across it. I'm sorry, I don't know whether that insulin is possible for that to be administered by transdermal route. You can give it by subcutaneous injection, but application on the skin, the peptide is too large for it to be absorbed through the skin. I'm sorry, I do not know the answer to that one, but I don't think it will work for it to even have a dose dumping.
Okay, sir. Sir, another question. What is the exact mechanism, including in microfluidic devices, as we have come across this phase very often these days? It could help if you elucidate the mechanism.
I'm sorry, I'm not very familiar with microfluidics, to be honest with you. So I'd be guessing the answer, so it's not worth.
Okay, can we avoid first pass metabolism by nanoparticles through oral route?
Again, where the drug is released from the nanoparticles, if the outer core of the nanoparticles is protected when given by oral route, then it has to go through further down the gastrointestinal tract, and then if it is released in the ileum area, then yes, definitely we have overcome that. But the main problem is it is nanoparticles. You're giving it by oral route. It has to go through the brush borders of, first it has to go through the gastric epithelium, then it has to go through the brush borders that you got along the duodenum and further down the gastrointestinal tract. It's highly unlikely that you'll be able to get the nanoparticles all the way down unless you put these nanoparticles in another capsule, which can protect these nanoparticles in these conditions. So there's further formulation that would be required, I would say.
Yes, sir. Yeah. How can we give corticosteroids either by oral route or parenteral route?
Corticosteroids are given orally also. Budesonide is available orally. Parenteral route is also available. Hydrocortisone injection is also available. It can be given by both.
Yes, sir. Is epidermal drug delivery is possible through injectables rather than topical agents?
Through the epidermis. Well, basically, you've got to go through the stratum corneum. What is it? That's going through the microneedles. Even you can reduce them. You can so you're basically going past the epidermis. But epidermis is actually made of 5 layers, and one of the main layers that you got is the stratum corneum, which prevents the absorption of the drugs or anything for that matter, because that is a protective barrier that every living being has got. So if you are able to overcome that barrier, then yes, definitely we'll be able to do that.
Okay, sir. Another one, sir. Can we make the drug pass through the blood-brain barrier by using nanoparticle drug delivery?
Fantastic. That's what I am working on currently. And we have developed a system using liposomes and gold nanoparticles. In this particular case, the liposomes, which have got a positively charged ligand and a negatively charged lipid. So you've got ionic interactions. So, and that's the gold nanoparticles as well, which are incorporated within it. So the liposome with the drug, which is incorporated in there, which is the opposite charge, and it goes past the blood-brain barrier, and that's where it dissociates and releases the drug. Because they are simple lipids, they just get broken down and then excreted. So you do not have any localized toxicity. Yes, it is definitely possible, and that is something which my team is working on.
Yes, sir. What are the benefits of liquid embolics? Liquid embolics.
Not sure I understand the question.
Okay. Okay, sir. Oh. How NEDs play a role in combating COVID?
How what plays a role?
Novel drug delivery systems.
Novel drug delivery.
Yeah.
Okay. Well, it depends on the drug again. I mean, we do have the antiviral drugs which are being tried out, like remdesivir and others. But I don't think we've looked at new delivery systems. We're looking at the box standard ones at the moment. And I'm sure in future something will come out with a nanotechnology or microtechnology based, or even smart technology based drug delivery systems too, for delivering these antiviral drugs. Not necessarily just for COVID, in general for antivirals, I'm saying.
Yes, sir. Few general questions, sir.
Mm-hmm.
what are the career opportunities in UK after B Farm or PharmD?
That is something which I work on. I am the course director or course leader for Overseas Pharmacy Accreditation Program. So if you are a pharmacy graduate from India or from anywhere outside of UK or European Union, then when you come to UK, you have to do a conversion course, which is for 1 year. And that particular course will help you come up to speed with the legal aspects and the clinical aspects that a pharmacist needs to follow if they want to practice as a pharmacist in the UK. So what we do is we have a series of modules or subjects which we teach. So we bring them up to speed, and also clinical placements which help them understand about how to deal with patients, how to talk to patients, how to prescribe, how to dispense the drugs, etc. All these factors. Once they have finished this course, once a student finishes, then they go through 1 year of pre-registration training, either in a hospital or in a community pharmacy. And once they finish this particular training, they go through a separate test, which is conducted by the General Pharmaceutical Council, which is the professional body, which is similar to our Pharmaceutical Association, Indian Pharmaceutical Association. So they conduct a test, and once you clear the test, you'll be given a membership of the Royal Pharmaceutical Society. That means you are a registered pharmacist. Once you're a registered pharmacist, you can work in either retail pharmacy or in a community pharmacy, or in a GP surgery, or in the hospital as a pharmacist. So that's the straightforward pharmacist route. That's either for a BPharm or a PharmD. It's the same route you have to follow.
Okay, sir. Another question, sir. Is our MPharm degree recognized there?
Yes, mine was.
Okay.
So, and for what? Basically, you either for working or for doing further studies. If you're doing further studies, then you can go. If you've done an MPharm, you can directly enter into a PhD course in UK. If you have not done MPharm, if you've done only BPharm, then when you come to UK, you have to do 1 year of master's course, any master's which is related, and then you go into the PhD. So in India, if you're trying to do MPharm, it is for 2 years. Here, if it is MSc, which is what we call, then that is only for a year, and then you go directly into a PhD. But again, of course, you have to reapply for a PhD and then go through.
Okay. Another one, sir. Can BPharm graduate will enter into research field?
Not as a graduate. I wouldn't take even a undergraduate, even from a graduate from our university, even, because they can only go so far as in terms of research. They'll only be a pair of hands. But if you want to do a proper research, then further studies would be recommended.
Sir, another one, how to select research proposal for PhD? Are there any sources?
There are two options here. One is based on your interest, which will be great if you are able to get it. Okay, if you are able to generate that, you can do that by say you have done a master, you have done a MPharm, so you have possibly done a particular project on it, and you would like to explore that further. So you would know what the future work is. So you're expanding on your training, all that you already have. So and then you go further, and then you write a proposal which is based on that. That way, what happens is when you apply for a grant from any of the grant giving bodies such as the DBT or DST or any of the other places, or even Commonwealth Scholarship, they will look at it favorably. The second way is where the supervisors or the guides, they would have got funds from the from these various funders, and they would have be working on a particular proposal, and they'll say, I want somebody, a PhD student, to explore this particular question more in detail over the next 3 years. So in that way, what you're looking at is somebody else's idea, but your contribution to actually explore that question. That goes back to saying it doesn't have to be a positive answer at the end. Even a negative answer is also a good answer, because that actually disproves the hypothesis which was originally written. So that would be the other route. So these are the two main things that I am aware of how students usually apply to do their PhDs with me.
Yes, sir. One more question, sir. Which route of administration is best when we have less bioavailability for inhalation route than oral route?
So you got less bioavailable by inhalation.
Inhalation.
Yeah, it is. That is, it's known, because major problem with inhalation is you have to go through a certain aspect. You got the three main ones: the powder dose inhaler or the metered dose inhalers, and the nebulizer. Nebulizer aside, for these two, for the PMDIs and the DPIs, the main problem comes with the cough reflux. So only about 10% of the drug actually gets down into the lungs. So you have a very poor administration of the drug reaching the site of action anyway. And on top of that, you need that to be active. So that actually is a challenge. So if you give it orally, then again, if your drug is got very narrow therapeutic index, for example, let us take theophylline. Theophylline is the third stage treatment for asthma. So it's not the first line of treatment. It's only in the third stage that you would give it. Main reason being it's got a very, very narrow therapeutic index. So why can't we give it by inhalation route? Again, it's highly water soluble. So it is not easy for the drug to be given by inhalation and for that to be absorbed. So depending on the drug, you choose you design your dosage form.
Okay, sir. Mostly this was the last question, I think, sir. Can you give more information on zeta potential?
On zeta potential, zeta potential basically means it's the surface charge that you have. Say, for example, you have the outer shell of the polymer, which is made of polylactic acid. It is lactic acid, so it's a carboxylate. It is COOH. So it is in an ionized group, it is going to be COO minus. So the surface charge is going to be negative based on the functional group that is present. The reason why you need to pay particular attention for on the surface charge is especially when you're preparing a suspension formulation. Because if you have like charges next to each other, they're repelled. So you will have the suspension, which is more stable and more uniform, the particles more uniformly dispersed. So that's basically, in a nutshell, what the zeta potential would be.
Yes, sir. So those are the questions posted in YouTube as well as Zoom chat box, sir.
Okay.
Yeah. If any of the participants want to interact with sir, please raise your hand. Our technical team will unmute you.
Yeah, very good evening, sir. Very good evening, sir.
Good evening.
Yeah, this is Dr. Ramaya, Associate Professor and Head of the Department of Pharmacognosy and Phytochemistry.
Okay.
Yeah, thank you, sir. You are enlightened with all the details of curcumin and all. So I have small doubts, sir, out of my curiosity. So, sir, though many studies mentioned on curcumin, in clinical trials it has not been successful. And also in 2017, US FDA what states that so curcumin is a fake cancer cure, right? So is the statement given in 2017. May I know the what's the reason behind?
Okay. I work extensively on curcumin, so I can answer some of it.
Yeah.
The main reason for if you look at the clinical trials, it's not just one. There are plenty of clinical trials that have happened on curcumin. During my talk, I mentioned that you have to give something like 12 grams, even after you give something like 12 grams of curcumin, you only have about 11 nanograms of the drug being detected in the plasma. Excuse me for 1 second. Sorry about that. So you have a very, very low bioavailability. The main reason is the formulation has not been designed in such a way so that you can actually increase the bioavailability of the drug. Two, if the formulation itself, if the drug itself is not soluble in water or in oil so easily, then proving or disproving its activity is extremely difficult. Having said all these things, it has been around for thousands of years. We still use it in cooking, and it has actually shown really good activity. I mean, for myself, I've actually tested out its anti-inflammatory activity. This is way back in 1992 by giving it orally, comparing it to the drug that the structural analog that I synthesized. And we've shown that just giving it as a suspension, it actually managed to reduce its reduce the inflammation that that has caused that we have given to the animal model. As far as the anti-cancer activity goes, it depends. Anti-cancer is not an easier, or rather, it's not an easy thing to assess for. The main thing is every cancer presents itself in a different way. No one knows how it actually starts, and it is not possible, or it is very difficult to give cancer to a normal cell in vitro without using any carcinogen or without using some other cancer cells and giving it to it. So it's when you do not understand the disease itself of how it is starting, and then trying to look at the drug and how, whether it is good or not, is actually quite a difficult statement, or a difficult thing to prove or disprove, I would say. I do not agree with the FDA's statement saying that it is a fake cancer, but maybe not. Well, if it might not be an anti-cancer drug, that's definitely not what I was looking for when we looked at tamoxifen. What we were combination with tamoxifen, what we were looking at is reducing the toxicity of these anti-cancer drugs on other cells when we give it. So curcumin might be able to protect the other normal cells around it while increasing the anti-cancer activity of the actual drug itself. So maybe that might be one aspect which, if it was really cytotoxic, giving 12 grams definitely should finish people off. So I don't think that is the case. So that's my opinion, by the way.
Yeah. Thank you, sir.
No problem.
Any other participant want to interact? Please raise your hand.
I request our technical team to unmute Dr. Nagabhushanam garu.
Good evening, sir.
Yeah, good evening.
Sir, myself, Professor M.V. Nagabhushanam. I'm a alumnus of BITS Pilani and working here as HOD and Professor of Regulatory Affairs Department, sir.
Oh, nice.
Yeah, nice to hear to you, and we are fortunate enough to have a blessings of yours with our students. Sir, I have guided 5 PhDs so far, and all of us have worked on cyclodextrins for enhancement of bioavailability of BCS class 2 drugs.
Mhm.
And a combination of various cyclodextrins along with hydrophilic polymers were tried for enhancement of bioavailability of these poorly soluble drugs. And mostly my PhDs steroidal anti-inflammatory drugs only for improvement of their bioavailability. Can any of these techniques be used for improvement of bioavailability of curcumin, sir?
I have tested out
Sir.
the beta hydroxypropyl cyclodextrin,
Sir.
and I've also tested the gamma cyclodextrin as well,
Sir.
both to try and increase the water solubility of curcumin.
Sir.
And
Yeah.
it had been quite tricky in order to prepare the formulation.
Sir.
What we have done finally was looking at soft minute extrusion with combining the cyclodextrin, curcumin, Soluplus, and microcrystalline cellulose. So all of them blended together, and then extruding them, and then pelletizing them. That seemed to have increased the water solubility marginally compared to the curcumin alone. That is, we managed to get, while 11 nanograms per milliliter was the solubility of curcumin, we managed to increase it to about 25 micrograms. So it went up. It went up, but it wasn't sufficiently high that it can be given orally.
Yeah. Yes, sir. Yes.
So that's what I tested.
Thank you, sir. Thank you
No
very much.
Any other participant? You please raise your hands. Sir, meanwhile, we got another question, sir. Levofloxacin disintegrates in stomach, maybe degrades in stomach. Can we make it colon specific by using enteric coating?
It can be encapsulated, and then the capsule can be enteric coated. That's possible.
Okay.
But you need to look at the stability. If you're trying to make it into an enteric coated tablet, you need to look at the stability of the antibiotic itself.
Okay, sir. Another question, sir. Compared to lupicillin and Baslock, which is more effective to the body?
I don't know what these two are. Sorry, I have no idea. I have no idea what they are. Lupicillin.
Yeah. So if you want to interact, please raise your hand. Otherwise, we'll end the question and answer session. Okay, sir. Thank you very much for your nice explanation and patient reply to all the queries, sir. Thank you very much.
Pleasure. Thank you.
Thank you so much, sir. enlightening us, clarifying the doubts in a full manner. I'm glad to inform you that more than 500 participants have attended through Zoom as well as YouTube Live.
Wow,
I
that's a
hope
that's a big number.
I hope everyone has acquired enough knowledge through this session.
I'm sorry, Dr. Sunita, I think you're muted.
Yes, sir.
Yeah, sorry. Yeah.
Thank you so much, sir, for sparing your valuable time with
No
us
problem,
to
pleasure.
clarify the doubts and enlighten us by sharing the innovative tips to focus on latest advances in LEDs. We treasure your valuable interaction with us and hope this program paid off handsomely with useful and profitable ideas. We look forward to seeing you next time. Thank you so much, sir.
Definitely. Definitely. Thank you very much.
I'm glad to see Dr. Aparna Kakarla garu. So, hi, hello, madam. How are you? So, if you permit, I would like to invite you to address the gathering, madam. Aparna Kakarla. Hi. Yes.
Hello everyone. I'm a clinician and I work in family medicine. I've been listening to the talk. It had been very, very interesting. So thank you for inviting me. I'm not quite sure what you would like to know from me, but listening to all the conversations and the questions, it's very encouraging to see young people being so much interested in research and with research ideas. It's very interesting to see the other side of the game. We only get the medicines and we work on it. So it's nice to see what happens and the thought processes that go through in making a medicine and the different ideas that get generated out of all these intelligent people. From my point of view, all I can say is as clinicians, we are heavily reliant on pharmacists in UK, not only for advice, whether it's in terms of drug interactions or usefulness of a particular medication. We also dependent heavily on them for clinical care. So for students who were asking Dr. Panala earlier about the role of pharmacists and the career prospects in UK, I just want to add that we are retraining some of our pharmacists so that more and more clinical care can be offered, because we have a lot of shortage of doctors. So clinical pharmacists are gaining a lot of importance. And clinical pharmacists are not just checking blood pressure and pulse rate. They're doing medication reviews, asthma reviews. We have a lot of people who are dependent on opioids and benzodiazepines. So for such people, we have our pharmacists who take great care, monitor them regularly. We have a lot of patients who are on cancer drugs, on antidepressants, some controlled drugs as well. And again, our pharmacists are doing an excellent job in monitoring these patients. In addition to that, the other thing they are doing a lot is monitoring patients with regards to drug, the DMRDs, which are disease-modifying drugs like hydroxychloroquine, leflunomide, methotrexate, so on and so forth. So long and short of it is, I think what is important to remember is we are no longer individual professionals. We are all interprofessionally related. We need doctors, we need pharmacists, and we cannot live without each other. Gone are the days when it was, I tell you and you listen to me. It's not like that anymore. We have a different approach these days, and that's how it should be. I think we need to learn off each other I have my special skills, you have your special skills, and together I think we can make we can provide a better clinical care for the patients. That's all I wanted to say.
Thank you. Thank you so much, madam. We have
Actually, doctor, I think Aparna will be able to answer this question, which somebody has asked about lupicillin and Baslog, which is more effective in the body.
I have
Both
no
are-
idea.
Both are insulins.
Well, I have no idea because you cannot paint the brush. We cannot paint everyone with
Yeah.
the same brush. So it has to be individualized. So what works for one patient doesn't necessarily work for the other patient. So I think it would be unfair to say one works better than the other.
Thank you, Dr. K.
okay. Madam, so nice to hear you, madam. Not only pharmacists, we are offering here Pharma D course also. Many of them are Pharma D participants are there. So can you suggest for them what are the opportunities are available at UK? For
I think Adant is probably better placed than I am to answer that question.
The PharmD students also have to go through the same thing, which I mentioned earlier about doing the conversion course for 1 year, and then they have to go through the pre-reg for 1 year, and then they can become registered pharmacists. So it's the main aspect for if anybody either a BPharm graduate, MPharm graduate, or a PharmD graduate, if they want to work as pharmacists in the UK, then they have to go through this 1 year conversion course called the Overseas Pharmacy Accreditation Program. So I'm the course director. We run the course. There are only about 7 or 8 universities in the UK that run this particular course, and we are one of them. Brighton is one of them, and I deal with it. So we train the students in that 1 year on clinical aspects, on community, on counseling, on dispensing, and all these various factors which we expect the pharmacist to do. And the main aspect is being able to do some of the clinical things, which, as Aparna was saying, that we make sure that they are aware of it. We do have what is we call a sim man. So it's a simulated man where it's a robot where we can give any disease to that, any condition to that particular robot, and the pharmacist is supposed to treat them straight away. So be it hypertension or dry eyes or asthma or any of these conditions, and then so we do train them like that. And once they go through that 1 year, then they have to go through the GPHC pre-registration and then the GPHC exam in order to become a registered pharmacist.
We do offer, so in our practice, we do offer some of these overseas pharmacy students come to do a clinical placement for half a day. And they have the experience of sitting with the doctor to see the kind of medication reviews you do on a daily basis. But also they get an opportunity to sit with the clinical pharmacist to understand the role of a clinical pharmacist. It's not about saying, are you taking the medicine? Okay, fine, go away. It's not just that. It is trying to understand the compliance, understanding, making sure the patient understands why they take the medications, communication, effective communication, and also picking up on problems which perhaps if they are beyond their remit of their expertise, it's knowing who to refer to or when to refer to or whom to contact. So we do offer placements in our practice where some of these students have found it quite valuable.
Okay. Thank you, madam. Now I request Dr. Rajkumar Garu to present citation for the speaker. Sir, we are unable to hear you, Rajkumar Garu. No. So please set your microphone once again, sir. Meanwhile, no, sir. No, sir. Again, meanwhile, you please try. And now I request.
I think you're.
Now. Now I request a man with a great vision, highly knowledgeable, focused, a strategic leader, and has a passion for excellence. As a pioneer of this institution, his care and love has nurtured this college to reach its dizzy heights of success, none other than our secretary and correspondent, Dr. S. Madhusudan Rao Garu, to express his opinion of today's program. Yes, sir. Sir.
Sir, am I audible, sir?
Yes, yes, sir. Sir, please go ahead, sir. Sir. Sir, we are unable to hear you.
I'm not able or what?
While questioning you. No, it's partly, sir. It's audible. Again, it's disconnecting. Sir, ikkade unnare madbettandi. Sir. Sorry for the interruption. Now I request, sir. No, sir, no.
Sir Rajkumar, sir, you are also not audible, sir.
Okay, sorry for the interruption. Meanwhile, we'll. Sir. Now I request our Honorable Secretary and Correspondent, Dr. S. Madhusudan Rao Garu, to express his opinion. No, sir. There is a poor net connection, I think so. We are unable to hear you. Sir, please unmute.
Somebody has to unmute. Please, Madhusudan Garu, మీరు unmute చేయండి.
Sir is having a poor net connection. So, okay.
But his mic is mute.
Sir, we will rectify that problem. Meanwhile, now I request our principal, Dr. Gondarajan, to give his message.
Audible? So on outside, I'm express my gratitude to all the organizers. On behalf of management, Sri Jubi Rangaraj Garu, Chairman Sir, and Dr. Masunara Garu, Secretary and Correspondent, and also Shankar Prasad Garu, and today's speaker, wonderful sessions which he heard to given enriching the knowledge to all the people who are there in doing research, who are budding pharmacists, and who are there want to do in future career. Really, sir, are expected Dr. Panala sir, and also Dr. Aparna madam given good information about what is clinicians are doing in the UK, and also the senior faculty members who are joined here, and I'm really thanks to given the wonderful opportunity to bear my view, you sir. So this our college is running various courses, but students still having the dynamo of how to choose the right career and how to choose the right path to do the research. As your wonderful experience, it shows that you are, as a scientist and followed by your good teacher, you are given enormous the brightening of the particular topics, which is good in curcumin. Really, we are in our department. I am the professor in department of chemistry. We are also doing the curcumin and ubiquinone to touching the antioxidant and also to derive various structure modification also we are doing. But still, we are given good support to us and enlightening the how to take the career. So really thankful, sir, and given good sessions. We are all enjoyed, and also we invited outsiders. So many senior professors also we invited this session, not only our students, sir. So we want to further your support to make it our college. We collaborated with your institutions to get some good kind of research. We want to take it with your support. Really thanks, sir. You came here and given wonderful session, sir. Thank you, Ananda, sir.
Thank you very much,
sir.
Thank you.
Thank you, Dr. Govindarajan. Sir, I would like to ask you, by seeing your profile, your specialization is in chemistry, but you completely deviated and expertised in pharmaceutics. What's the reason, and how did you change it, shifted to from one area to another area?
I haven't shifted. I would say I haven't shifted. The main reason I'd say is if you don't understand chemistry, you cannot do anything. It doesn't matter whether it is pharmaceutics or if it is anything else. Pharmaceutics is just another subdivision of chemistry, according to me. Okay. You need to understand the drug, which is chemistry. You need to understand the excipients, which are again chemical compounds. You need to know what is the interaction that is happening. That is all based. I'm just looking at more of application basis of chemistry rather than looking at pharmaceutics. I'm going to look at tablets only. So I'm not. So this is some. Even when I was working in the industry, that's where I had realized when I was preparing the new formulations for these new drugs which are there. It is that you have to understand the chemistry of the molecule before you can actually do anything. If you don't understand the chemistry, you won't be able to prepare a natural, proper formulation. So that's where I started, and that's so that's my opinion. I think it is still the same. I'm looking at more of application aspects, and it might be interesting to know we don't have a pharmaceutics department in our university, in our school.
At that time,
in our pharmacy school, we don't have a university pharmaceutics department. It is pharmaceutical sciences, pharmaceutical and chemical sciences. So everything which is medicinal chemistry, pharmaceutics, analysis, all of them come under the same branch,
Okay.
including regulatory affairs.
So you are the role model to our students and upcoming generations, sir, because generally we are concise to our specialization in a particular area, but that is not the correct way. In future, we have to acquire much knowledge in a broader area, irrespective of our specialization. You have proven in that aspect. So I
Thank you.
felt so happy you are expert in all the areas, not only physics and chemistry. Thank you so much, sir. Now I request Dr. Rajkumar Gar. Are you ready?
Madam, am I audible?
Yes. Now I request our Professor Dr. K. Rajkumar Garu, Department of Pharmaceutical Analysis, to present citation for the speaker.
Thank you, madam. Good evening, sir. Dr. Anand Pannala, sir. I am Dr. Rajkumar Kudari. Nice to hear from you that you got graduated your BPharm from Manipal in 1991. I feel happy to say that I did my BPharm from same Mangalore University and got graduated in the same year.
Oh, okay.
Yes, in
From Mangalore.
1991.
Okay.
From Nitte
From
College,
Nitte.
NGS.
Yeah.
And Dr. Satyanarayana sir was our principal then.
Yeah.
DS sir, who was the first batch student of Manipal.
Yes.
Here is a citation for you from Hindu College of Pharmacy, sir. Though it is not the right meter to measure your excellence, but still, with our little attempt, with all our efforts, we prepared an incredible journey. It goes like this. Hindu College of Pharmacy, Guntur, AP, India. Citation, Dr. Anand S.V. Pannala, MPharm, PhD, FRSC, SFHEA. An incredible journey. We fail in words to express the altitude of our appreciation for the amazing and multifaceted work that you have done and are doing in the twin fields of academics and research in pharmaceutical sciences at international level. Your research interests especially are awe-inspiring in their variety and applications. Starting with the auspicious curcumin at your master's level, you have scaled new heights in industrial pharmacy, NDDS, encapsulation technology, NCEs, nano formulations, targeted drug delivery systems, etc. The list obviously is endless. The unique fingerprints of your research on budesonide, theophylline, liposomes, nanoparticles, column interactions, and gold as a chemical compound, being though it is deviated, droplet technology in agriculture as well as biology, and various other pharmaceutical preparations bear ample testimony to your exemplary capabilities in research. That you are equally competent and being well placed in academic field is also evident by your different and tailor-made approaches to teaching and learning, and by your nurturing with scores to students for PhD and postdoc levels in your own mold. Thomas Alva Edison once said, if we did all the things we are capable of, we would literally astound ourselves. Perhaps the day is not far off, sir, when you are astounded by your own gigantic contribution to both academics and research in UK and back in India too. We are privileged and also proud to have you with us, sir, as a great source of inspiration for years to come in this incredible journey towards the summit of perfection. Thank you, Sri Jupudi Rangaraju, Chairman, Dr. S. Madhusudan Rao, Secretary and Correspondent, Dr. Govindarajan R. Principal, Dr. A. Sunita, Vice Principal, faculty and students. A submission with due respects to Dr. Anant SV Annala.
Thank you very much.
So this will be sent through Shankar Prasad sir through your email or the means that we can find out. Once again, thank you, sir. And
Thank you very much. Thank you.
I'm very happy that we both are contemporary and from the
Yes.
same university.
From the same university as well. Yes.
I did my M.Pharm in analysis, sir, and I'm working as professor here at Hindu College, having 12
Okay.
years of teaching
Oh, that's
experience.
very nice to know. That's very nice to know.
Thank you, sir.
Thank you.
Over to Sunita, madam, our
Yeah.
convener.
Thank you, Dr. Rajkumar, sir, which I have not covered while introducing the today's speaker. I felt so happy. We fulfilled sir's profile. Sir, please accept our citation. We will forward a mail to you
Definitely.
today itself.
Definitely.
Now I
Thank you.
request a man with a great vision, highly knowledgeable, focused, and has a passion for excellence as a fine.
Just is enough. Yeah, enough. Good evening,
Sir.
sir. Good evening.
Good evening.
Dr. Shankar Prasad, very good evening. I owe a lot to Shankar Prasad for having introduced you to us, because I just wonder, our association has been more than a couple of years, 3 years. I wonder how we have missed you so far. I have not been introduced to us.
No, but he knows about the college, sir. He knows about the college on a day-to-day basis.
It's a mind-blowing and phenomenal career and profile. We have very badly missed you, sir.
Thank you. It's my
Perhaps
pleasure. My honor.
Manipal University and Manipal College of Pharmacy Business is very, very blessed to have an alumnus of your stature and a visiting professor of your stature at the same time. Now, I wanted to call you a Sabyasachi, being in teaching as well as academics, teaching as well as research in equal competence, just as our Dr. Rajkumar has read right now. Your career graph has been phenomenal. From TTK to University of London to Brighton. Really, it's rather a career which everybody would be envious about. And we are very fortunate and lucky. When I got your profile of 10 pages, I was just reeling with confusion what to convey to our faculty and what not. It's a great journey, sir. Incredible journey. And I am happy that we are with you today with so many questions and answered, and so much of information handed over to our students and faculty also. Shankar Prasad, thank you so much. And I promised from him that we will be meeting very often, quite often, about your rather guiding us in our future endeavors. So, sir, as our chairman has said and our convener, Dr. Sunita, has said, you are most welcome. Anytime you are coming back to India, once you please visit our college.
Definitely. Thank you very much.
When-
It would be my honor.
In India, we would come and meet you and pick you up from there. So it's a great day for us today. We have a lot of gaps to be filled in the college also, gaps to be filled, especially in research. As our principal has rightly said, you're being a chemistry man, basically, and teaching all the sciences at a time, that's a real wonder, because we have specialization for everything, Sutex, analysis, Gnosy, as well as chemistry, like that. But it really, I was just wondering how you could teach Sutex and analysis at the same time and chemistry also. There's a real opportunity for a man of your stature to do so many things at a time. You have so many accomplishments, so many affiliations, so much of love for your students, bringing them up and then seeing them in your own stature and in your own personality. That's a great achievement for a man. In a matter of just 2 decades, you have achieved a lot, which is rather mind-blowing. So, sir, thank you so much. Yeah, we meet once again. As Shankar Prasad is very close to us, probably we meet very often, at least through email or phone or whatever it is. And then I request you to please guide us, keep guiding us. Thank you so much. Thank you very much.
Thank you very much for your kind words, sir.
Thank you.
Thank you, sir, for your words of appreciation about today's speaker and the program.
I must thank Shankar Prasad Garu for sparing so much of time, patiently listening to all of us and then sitting through. Thank you.
Now I request our Pharma D Director, Sri Sita Rame Garu, to express his opinion.
Is he informed? Is he there? Okay, let him join.
We are unable to hear you, sir. Please check your microphone. Yeah, meanwhile, I request our special guest, Sri Shankar Prasad Garu, Pharma Scientific Advisor to Government of Andhra Pradesh and Punjab, give his benedictions, sir. Shankar Prasad Garu.
Yeah, madam. Sorry.
Sir, please.
Thank you all for providing a great platform to all the students. So whatever little bit possible help is from our side, we are trying to help you. But first, I would like to entire the faculty and the students should be thankful to management. As I told you, I'm going on in the recent days. I'm going to so many institutions, but I didn't find the people of such passion. Whether you were chairman or you were the secretary, always whenever I was there, they always say that they want to do something for the students. And what one way that has posed to me, whatever possible, little bit possible, I'm trying to do that. And yeah, there's a little bit late is there in getting Anand here. But for everything, I believe for everything, time has to come up. I think now time has come up. Now we will start it out, whatever the way best is possible. I think I can rest assured from his side, whatever help is required.
Thank
you. To do that. Thank you very much.
Thank you. Thank you, Prasad.
Thank you, sir. Without your support, we couldn't achieve and conduct such a wonderful programs. Thank you for your constant support and encouraging us to interact with many more eminent personalities in future also.
Certainly, madam. I'll do my best.
Thank you, sir. Now I request our Pharma D Director, Sri Sita Rame Garu, to give his benedictions.
Hello. Good evening, sir. Dr. Anant Pannala Garu. We are very happy to have your webinar today. Sir, we have B Pharmacy, M Pharmacy, Pharma D, and Diploma in Pharmacy in our college. And 54 faculty members are there. After them, 10 are PhDs. They have got 25 students who are doing PhD under them. And most of the students, both scholars, M.Pharm, B.Pharm, PharmD, are all listening this webinar. Not only our students, some of the neighboring colleges students from Acharya Nagarjuna University and other universities are also listening. I'm sure they will all be benefited by your lecture, which is given by you today. Well, these drugs, the novel drug deliveries, some of them have become very popular. As you have rightly said, those who are acting on the respiratory system and pulmonary system, they have become very popular. And some drugs like Nitrodam TTS, transdermal systems are also getting popularity. And I wish that those work which you have stated, the development of novel drugs in statins and in anti-cancer drugs, they will also, in due course of time, into the field because of their advantages. We can save a lot of drugs. Quantity of the drugs will be less, and their toxicities will be less. Ease of administration will be there. So, in view of all these advantages, these novel drugs will get a lot of demand in future, and our students will get inspiration not only from your speech, but also your experience, teaching experience, research experience, industrial experience. So in this connection, I would like to thank you, sir. Thank you very much. And not only you, but the one who has introduced you, Dr. Shankar Prasad Garu, our management, and our chairman, Rangaraj Garu, and secretary correspondent, Dr. Madhusudan Rao Garu, our principal, Dr. Govindarajan, vice principal, Dr. Sunita, Dr. Sita Devi, and others who have done their best to make it excellent presentation today. Thank you, sir. Thank you very much.
Thank you very much, sir.
Thank you, Sitaramaya Garu. Now it's going to end the session. I request Dr. K. Ravi Kumar to propose vote of thanks.
Thank you, madam. So, good evening, everyone.
Yeah, please proceed.
So, good evening, everyone. I am honored and lucky to propose vote of thanks on this international webinar session. Gratitude is not only the greatest of virtues, but the parent of all the others. So, first of all, I am grateful to Hindu College of Pharmacy Chairman, Sri Jupudi Rangaraj Garu, for his thought-provoking message. Thank you, sir. Let me express my gratitude to our Secretary and Correspondent, Dr. S. Madhusudan Rao Garu, for his remarkable endeavor. Our words may not be capable of communicating our sense of gratitude, and may I take this occasion to salute him with a deep sense of gratitude to keynote speaker, Dr. Ananta S.V. Pannala Garu, for the way in which he explained the advances in novel drug delivery system and trials was in an extraordinary fashion. So thank you very much, sir. So we offer our sincere thanks to Sri S.S. Prasad Madhiraj Garu for his inspiring words and his support, but for which the webinar would not have taken place. I thank Dr. Aparna Kakarla Garu for sharing valuable information and career prospects. I also wish to express my gratitude to Principal Dr. R. Govindarajan Garu for his message. I extend my thanks to Vice Principal Dr. A. Sunita Garu for comparing the program. I would like to thank OSD and PharmaD Director Sri P. Sitaramaya Garu for his informative speech. We offer our sincere thanks to our faculty members for their continuous cooperation for the success of the webinar. I would like to thank all the organizing team members for this webinar session. I must not forget to thank all the students for making this session a grand success. Finally, I would like to thank you again for making this webinar a very grand success. Thank you all.
Thank you, Dr. Ravi.
Suggestion.
Sir.
Madam, you are getting a different section of people from across the globe. All are expertise in different field. If you can constitute a small group so that whatever the research, whatever the current programs are going on across the globe, the one thing is what I have seen is that we have to update the students on the current research, what is going on across the world. That is what required. For which let's try to create a platform so that we will be, from faculty side, we will be able to update all the students whatever is happening in the pharmacy world. Yeah, thank you.
Thank you, sir. Hereby end the meeting by playing national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे, तव शुभ आशीष मागे, गाहे तव जय गाथा। जन गण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय जय जय